Hi everyone. Today we will discuss bacterial culture media, their types and applications. A culture medium is a mixture of substances that supports the growth and multiplication of microorganisms in the lab, or it is used for transport and storage. We can't imagine a modern microbiology lab without culture media. But do you know the name of the scientist who actually developed it for the first time and why? Although Leeuwenhoek observed microorganisms under his microscope, but the progress in this field was stopped for 200 years. The second revolution came with the discovery of culture media, which allowed it for the easy detection and characterization of microbes from the various sources. You might be familiar with Louis Pasteur, famous for his swan neck experiments that disprove abiogenesis. In 1860, Pasteur successfully prepared a liquid culture medium sufficient to grow microorganisms in the laboratory. The key success was the medium's composition was known and reproducible. However, to isolate a target microorganism from a mixed microbial population, it is necessary to grow them on a surface of a solid medium. This allows easy identification and characterization of each microorganism present and their proportions. I am sure you have heard of Robert Koch, a doctor who established the germ theory of disease, showing that the microbes are responsible for diseases. He tried to isolate and study microorganisms present in patient samples in laboratory. Initially, he used aseptically cut potato slice to grow pathogenic microorganisms, but this method had some limitations, such as difficulty in sterilization and the presence of some pre-existing microbes. And seeking alternatives, he found that gelatin could solidify liquid medium also. However, this posed another problems. Some pathogenic bacteria could utilize gelatin and liquefy it. Finally, one of the Koch's colleagues, Fanny Hayes, discovered agar. Agar is a polysaccharide extract from marine algae, and it melts at 84 degrees centigrade and solidifies at 38 degrees centigrade. Agar is not utilized by bacteria, and making it an ideal solidifying agent. This same compound still used today to solidify growth media. That's why Robert Koch is known as the father of microbial culture media. Since then, numerous media have been formulated to meet specific requirements for various bacteria. Media are generally named according to the name of the scientist who actually formulated them and for the first time, like Lowenstein-Jensen agar, formulated by Lowenstein and later modified by Jensen for the isolation of Mycobacterium for TB patient samples. If any modification is made, it is generally mentioned, such as modified Thayer-Martin agar, which is used for the isolation of pathogenic Neisseria species. Media are also named according to the main components used in them, like brilliant green bile lactose broth, used to isolate coliform microorganism. Sometimes media are also named depending on what type of microorganism will actually grow on them, like Salmonella shigella agar or in short SS agar. The composition of the medium actually depends on what type of microorganism you want to grow. You can see the composition of one minimal medium for cyanobacteria. They are phototrophic and can synthesize all essential nutrients from inorganic material. However, bacteriological media generally contain one carbon source for energy production, like glucose, lactose, sucrose, etc. And some bacteria can use various organic and inorganic compounds as energy source, like ethanol, fatty acids, proteins, petroleum, naphthalene, or even plastic. Some bacteria require amino acids and vitamins, which are incorporated into the medium in form of peptone and yeast extract respectively. To maintain the pH and osmotic balance, salts are used, and they are also support growth as a source of inorganic compounds. At present, nearly 7,000 different types of media exist, and depending on the physiological state of the medium, they can be classified into three types: liquid, semi-solid, and solid media. In liquid media, no solidifying agents are used, and allowing the bacteria to grow freely in the solution, like luria or teri broth. In contrast, semi-solid and solid media use agar as a solidifying agent, with the concentration of agar being higher in solid media than in semi-solid media. The nutrient agar plates you have likely seen in your lab are an example of solid media, typically containing 1.5 to 2% agar. On the other hand, sulfide indole motility or SIM medium, which is used to test the motility of bacteria, contains a lower concentration of agar. usually around 0.2 to 2.5%. Initially, plant or animal tissues were used to prepare culture media. While the general composition of this media is known, but the exact concentration of the composition remain unknown. Let me explain this further. Suppose you add 1 gram of beef extract to 1000 ml of culture medium. You are aware that this extract contains amino acids and vitamins. But do you know the exact composition of the component? No. Conversely, if you add 0.5 grams of magnesium sulfate to 1000 ml of culture medium, it is straightforward to calculate the exact concentration of this ingredient. Therefore, depending on the chemical composition, culture media again classify into two types, defined and undefined medium. A defined medium is also known as chemically defined medium or synthetic medium, does not include any plant or animal tissues. The composition of a chemically defined medium is listed in this table. You can see glucose is used as energy source and carbon source, and salt are used as trace and tracer elements, and they also maintain osmotic balance and pH. This medium can selectively allow some bacteria to grow, but most pathogenic bacteria fail to grow in this medium because they have some additional requirements. These bacteria are called fastidious bacteria. To grow them artificially in the lab, different media are used, like nutrient agar. You can see the composition of this medium in this table. Here we know that yeast extract contains vitamin B, and it's a very good source of amino acids also, but the exact proportions are not known. That's why this medium is called undefined or basal or complex medium. It contains all necessary growth supplements, and because of that, nearly all bacteria, whether it's a pathogenic or non-pathogenic, can grow on it. And that's why this nutrient agar is called a universal growth medium. Some commonly used components in the complex media include peptone. It's a common ingredient, and it's a mixture of enzymatic digestion of protein. It provides small peptides that microorganism can use. An yeast extract, it is used to maintain the number of vitamins, coenzymes and nucleotides. Casein hydrolysate is made from milk protein. It contains many amino acids, and it is used to enrich culture media. As time passed, the growth and multiplication of pathogenic microbes in the lab were not restricted. Now scientists want to know which microbes are present in patient samples, or they want to grow particular microbes while stopping the growth of others. New media are being introduced according to these purposes. A few commonly used types of media are discussed here. Enriched medium contains special nutrients that allows the growth of a particular microorganism that might not otherwise be present in sufficient numbers to allow isolation or identification. Enrichment medium helps to multiply bacteria that are very rare in number compared to other bacteria. Without enrichment, these bacteria can't be isolated because the predominant bacteria will occupy the culture plate faster than the rare one. These media do not kill or stop the growth of other unwanted bacteria. The enrichment method was discovered by a soil scientist named Martinus Beijerinck. Using this technique, he was able to isolate and identify nitrogen-fixing symbiotic bacteria present inside the root nodule of some plants. This discovery was significant because those soil bacteria called rhizobium are important for increasing the soil nitrogen content. The most common enrichment media used frequently to isolate pathogenic bacteria are listed below. For example, 5 to 10% rabbit or horse blood is used to isolate Streptococcus species. When blood agar is heated slowly to 80 degrees centigrade, the RBC lies and the color of the medium changes. Hence, it is called chocolate agar. This medium is used to grow Neisseria and Haemophila species. Another example of enrichment medium is Lowenstein-Jensen media, which is used to isolate Mycobacterium tuberculosis from patient samples. Selective medium encourage the growth of some microorganisms but suppress the growth of others. Antibiotic, chemicals, or dyes are often used as selective agents that specifically kill some microbes but allow others to grow. For example, McConkey agar, which contains crystal violet and bile salts that selectively kill gram-positive bacteria but allow gram-negative bacteria to grow. In clinical microbiology lab, McConkey agar is a very useful and widely used selective medium for Enterobacteriaceae. Thiosulfate, citrate, bile salt sucrose agar, or in short TCBS. It is selective agar medium for isolating Vibrio cholerae. Differential medium has a constituent that cause an observable changes. in color or changes in pH in the medium when particular biochemical reaction occurs. These changes allow microbiologists to distinguish a certain type of colony from others growing on the same plate. Most of the time, a pH-sensitive dye is used that change its color when the pH of the medium changes due to the acid or alkali production by the microorganism. For example, the selective or inhibitory agents in EMB agar are the dyes eosin Y and methylene blue. Methylene blue inhibits gram-positive bacteria, while eosin changes color to a dark purple when the medium around the colony becomes acidic. Here, strong acidity produces a deep purple color colony with a green metallic stain like Escherichia coli, whereas less acidity may produce a brown-pink colorization of the colony. Blood agar is also used as a differential medium. Different species of Streptococcus slice RBC at different rates, and which helps to identify the different Streptococcus species. MacConkey agar is also a differential medium. It contains lactose and a pH indicator dye that is neutral red. Lactose fermenting bacteria utilize lactose and lower the pH of the agar below 6.8. That result in the appearance of red or pink color colonies. Non-lactose fermenting bacteria such as Salmonella, Proteus, Shigella, they can't utilize lactose, and this forms ammonia, and which rises the pH of the agar and leads to the formation of white or colorless colonies. Nowadays, a new medium has been introduced to the diagnostic market called chromogenic agar. It contains different energy sources and pH indicators for certain bacteria, and after the incubation, it allows easy identification of the bacterium. For example, the Candida chrome agar or UTI chrome agar. In UTI chrome agar, you can see the different type of bacteria produces different color colonies, which helps the technician to identify them very easily. Next, we will talk about transport medium. Transport media for microorganisms are essentially a buffer solution containing carbohydrates, peptones, and other nutrients, but not any growth factors. It is designed to preserve the viability of a bacteria during the transport without allowing them to multiply. It is generally used when the sampling area is far from the laboratory. One such medium, called the viral transport medium, was extensively used during COVID-19 patient sample collection. Another transport medium, called the Venkataraman Ramakrishnan medium, it is used to transport fecal samples containing Vibrio cholerae. Finally, we will discuss about anaerobic medium. As the name suggests, it is used for the isolation of anaerobic bacteria. One commonly used anaerobic medium is thioglycolate broth. Sodium thioglycolate is added to this medium as a reducing agent that supports the growth of anaerobes and microaerophilic microorganisms. I hope this video helps you to clear your concept regarding various bacteriological media. If you like this content, press like and share it, and don't forget to write your feedback in the comment section. Subscribe and press the bell icon for regular updates. Thank you for watching and supporting.